Hey y'all, Dr. G here. This video is going to be the first in a series that goes over the overall process of translation. Right, so as you know, translation is the process in the cell that uses the sequence of mRNA to guide the synthesis of proteins. Right, so the general idea here is that you have these molecules called aminoacyl tRNAs. Okay, this is the activated form of an amino acid that is used by the ribosome to build proteins. The idea here is that you have this ester linkage between a tRNA molecule and the amino acid. Another important thing to recognize is that in this form, you still have a reactive amino group. Okay, so it's the C terminus, the carboxylic acid, that is conjugated to the tRNA. So the general idea in how protein synthesis occurs is you have two tRNA molecules, two aminoacyl tRNAs, right? So tRNAs loaded with amino acids. They enter into the ribosome, the RNA-based catalyst that catalyzes this process, and the amino group on the incoming aminoacyl tRNA serves as a nucleophile. It attacks the carbonyl in the ester of the aminoacyl tRNA on the existing tRNA. So simply, we have nucleophilic attack, electrons flow up to make an intermediate, they flow back down to reform the pi bond, and the ester linkage is broken, releasing a tRNA and now creating a new peptide bond between the second amino acid and the first amino acid. Okay, so this first amino acid, the entire thing is transferred over to the second aminoacyl tRNA. Okay, so now we have this dipeptide, two amino acids covalently linked to the second tRNA. You now have a third aminoacyl tRNA that comes in. It serves as the nucleophile. Same thing, nucleophilic attack onto the carbonyl. Electrons flow up to make an intermediate, collapse back down, and the ester bond is broken, releasing the second tRNA, and now creating a tripeptide linked to the third tRNA. So the idea is that the growing chain continues to grow, and it's always linked up with one tRNA molecule, the most recent tRNA that is placed into the ribosome. Okay, the other important thing to notice here is that the way this works is the first amino acid put on, right? This first amino acid put on is always the N-terminal amino acid. So amino acid number 1 is always the N-terminal amino acid. The last amino acid that gets incorporated will always be the C-terminal amino acid. So this whole process happens on the ribosome. The ribosome is this massive complex that's made of 3 RNA chains and lots of proteins. Okay, it's broken up into 2 independent subunits. In E. coli, we call this the 30S and the 50S subunit, respectively, for the small subunit and the large subunit. This S label, it stands for Svedberg units, and it's just a way of categorizing or determining the overall size of a molecule. Okay, and in this image here, kind of the idea is this is the small subunit and this is the large subunit. And an important thing to notice is that the active site, the chemistry happens at the interface between these two subunits. And this idea will be reinforced later in these videos. Okay, so the mRNA, the template, is interacting at, actually like binds to the small subunit. The aminoacyl tRNAs bind to the large subunit. The growing peptide is present in the large subunit, like there's the peptide channel there. And the chemistry happens at the interface. The ribosome is the catalyst in translation, right? This is one example of not using amino acids, not using proteins to catalyze reactions, but instead life has evolved to use RNA molecules, structured RNAs, as catalysts. And it's interesting that this is the catalyst for the entire process of making proteins, right? So you remove RNA, you can't make proteins, and that's kind of neat. Okay, so it has three distinct binding sites: the A site, the P site, and the E site. And in this image, we're actually showing two of the three sites. And we'll be able to see these better later on. The incoming site is known as the aminoacyl site. The A site is the aminoacyl tRNA site. That's where the aminoacyl tRNA originally comes into. The P site stands for the peptidyl transfer site. Okay? And the new peptide bond is formed between the amino acids in the A site and P site. And then the idea is that this linkage right here is broken when this peptide bond is made. The entire growing peptide or growing protein is transferred over to the tRNA in the A site, and then it gets moved over to the P site. Right? But that's kind of jumping the gun, but kind of giving you a heads up of what we're going to see how this happens. Okay, there are three different types of RNA molecules that are involved. mRNA is the message, right? It is what codes for the sequence of amino acids. tRNA are the molecules that are linked up with the amino acid that bring the amino acid in, that essentially decode the sequence of mRNA, right? It is the intermediate that will allow the right amino acid sequence to correspond to an mRNA sequence. And rRNA is the ribosomal RNA. That's the RNA chains that build the ribosome. There are three of them. Okay, as I mentioned, tRNAs are the adapters. These are really important in making sure that the correct amino acid sequence gets built into the protein that corresponds to the code built by the mRNA message. These enzymes, aminoacyl tRNA synthase, we're going to see them abbreviated AARS, aminoacyl tRNA synthetase. They are the enzymes that are ultimately responsible for decoding. They are the enzymes that match the correct tRNA that presents a certain anticodon with the correct amino acid, right? So it's these enzymes that make darn sure the correct aminoacyl tRNA is present and can go into the ribosome to make proteins. And we can very much think of these aminoacyl tRNAs as the substrates in translation. The ribosome is the catalyst, the aminoacyl tRNAs are the substrates, and the growing peptide chain is the product. So let's think about the ribosome for a second here. It is absolutely massive. In E. coli, in basically all organisms, it consists of three chains. The small subunit is made up of 16S ribosomal RNA. The large subunit is made up of the 23S and the 5S. So the large subunit has two chains of RNA. The small subunit has one. And I mentioned before this S term. This just it's a label for Svedberg units, which is the unit of the sedimentation coefficients. It just tells you about how big something is. So the larger the number, the bigger the molecule. Okay. The other thing that's relevant here is that there are 52 proteins associated with the ribosome in E. coli, and even more in eukaryotes. Most of them are associated with the large subunit, but about 21 of them, pushing half, are associated with the small subunit. Okay, the really important thing to take home here is that these things are massive, so big you can see, right? You can see them in using a microscope, right? They are, when you think of like the rough endoplasmic reticulum, these are the little dots that dot the rough endoplasmic reticulum. They are massive complexes. Okay, another really, really cool thing about the ribosome is just how good it is. Right? There are very, very, very few mistakes. Okay, so we think about it in terms of this thing called the error frequency. And in E. coli, translation has an error frequency of around 10 to the minus fourth. So one mistake in about 10,000 tries. That's really awesome. And it's also really, really important, right? Because if we had more mistakes, then we would have then it would be really difficult to synthesize the correct sequences of proteins, right? And this chart I really, really like because it kind of gives you an idea of the likelihood of making an error-free protein given different error frequencies. So 10 to the minus second, meaning 1 in 100, even for a amino acid, right? So for 100 amino acid protein, we only have like a 36% chance of getting it right. When we get down to 10 to the minus fourth, we have about a 99% chance of getting it right. And if we were to get to 10 to the fifth, essentially 0 errors would be incorporated, even in a 1,000 amino acid protein. Right? So this idea of error frequency is important. And in all organisms, the process of translation has evolved in a way that minimizes errors. Okay, so this brings us to the idea of the genetic code, which we're going to take a little bit to explore. I'm very confident that you have looked into the genetic code before. So let's just think about it briefly. We know the general idea is that codons are built of 3 nucleic acids, position 1, position 2, and position 3. And those that sequence of nucleic acids will then code for a specific amino acid. And it's adapted by the tRNA, recognized by an anticodon, which creates a base pair with the codon. Okay? You can look at a really useful table that can tell you very quickly what amino acid is coded for by a specific codon. On the rows here, right, on our y-axis, this tells you the first letter. So everything in this row starts with a uracil. Everything in this row starts with a cytosine. The second letter are our columns. So this box is all UU. This box is all UC, etc. And then the last position is represented by each of these. Okay? So there's some really, really cool themes that come out of the genetic code. And one is the importance of redundancy. You'll note here that in many, many, many cases, one amino acid is coded for by lots of different codon pairs, right? So for example, proline here. Proline is coded for by any of the codons that start with CC. So any of them will code for proline. Same goes with arginine. Arginine, anything that starts with CG will code for arginine. And there's really, really important reasons for this that we're going to explore after you. And that brings us to a point where we can go ahead and work through the first bit of your activity. So question 1 in your activity focuses on thinking about the genetic code and trying to identify some important aspects of redundancy. So go ahead and take a few minutes and try to figure that out.